Okay, so I'd like to introduce this paper that published this month. Yes, really newly published the papers. Okay, please look at the title. Yes, so you can understand the keyword is the coronavirus. Yes, related to the SARS-CoV-2. Okay, but this virus was isolated from this animal, the pangolin. Do you know the animal pangolin? It's similar with armadillo. Yes, so please look at these pictures. Maybe you know that. Okay, okay. And anyway, in this paper also isolated the coronavirus from pangolin, and also they carried out experiment. What a kind of experiment? In laboratory, they try to culture these virus and also carry out the pathogen challenge test. It means that they infect the mouse with it, that virus. And they try to emphasize the result shows the lethal infection. It means the mouse infected with this virus died. That's why lethal infection. Another problem is this one. The mouse used in the experiment is not normal, not standard, because title said the human H2 transgenic mouse. So it means already the mouse we can call say humanized mouse. Why? Because the human AC2 it's a human gene already transferred into the mouse. So it means they isolated the new virus and they infect the mouse, very the specific mouse, human H2 transgenic mouse, with this virus, and the result showed lethal infection. All mice died. Okay, so please look at the abstract. Okay, yes, the SARS-CoV-2 related pangolin coronavirus can cause 100% mortality. 100% mortality. All mice died. And especially, as I said, this mouse is very specific because human H2 transgenic mouse. Okay, and H2 means this is a very important receptor for the coronavirus because coronavirus via this receptor H2 they can enter into the human bodies. This is one of the entrance for coronavirus. Yes, this must already contain this the receptor, and so new virus contacted this one and enter and kill all the mouse. That is very important things. Okay, and please move to the actual data. Okay, please look at this one. Okay, so I want to show the just a few figures. Please look at the figure B. This is the result of the survival of mouse after infection. X-axis means the days post-infection, Y-axis means the survival rate of mouse. Yes, and the case of the control, it means the dot line, and the stable, it means 100% all mouse the healthy and can survive. But a case of this one, in this the paper, they isolated two virus, coronavirus, and the name is REC7, and the other one is GXP2VC7. Yes, and in case of this one, second one, the pink line, as you can see, day 7, the drastically decreased, and after that, the 8 days, day 8, so all the mouse died. 100% died. And in that case, they knew the coronavirus, how they can enter into the mouse bodies. This, please look at the figure D, and the y-axis means the copy of the virus. It means the number of virus, and y-axis means each organ from left side like a heart, liver, spleen, lung, something like that. Okay, pre-focus on the lung and the brain. Yes, the copy of virus too high than other organs. And also, the bloom in after 3 days of infection, and the pink square means the after 6 days of infections, especially case of brain. So the down day three, the copy number is drastically higher than the in the day six. Day six, the copy number is very high than 6 days. So it means the after infection, this new coronavirus can enter into the brain and infect, and finally the kill all mouse. Okay, and also the figure of the F and G shows this is the microscopic observation of tissue. The left side is lung and the right side brain. The data showed, basically after infection of coronavirus, the construction of the tissue texture changed. But in this report, not so different among the control and infected mouse. Okay, so let's move to the bottom. Okay, and let's see, and please focus on the around here. Yes, here. Okay, especially in case of the coronavirus infections, there are some parameters affected, especially case of the inflammatory factors. So it means one of the cytokines like IFN gamma and IL-6, something like that. So basically, in case of infection by the coronavirus, these factors increase. It means that inflammation should be caused. But in this paper said, after infection in mouse, these parameters did not change significantly. So it means so a little bit different with the previous coronavirus. But anyway, data shows are newly isolated from pangolin killed all mouse based on infection in brain. Yes, and also I worry about that here. Let's go back to the figures. I said in lung and the brain, so the very higher the virus copy. But in addition to lung, and I found here turbinate also higher copy number than other organs. So it means, of course, a virus can enter the brain, but I think in lung and the turbinate virus stay there. So it means when we breathe air, so virus can go outside and move to other individuals. Please have an image. This is an experiment with mouse, but I said the mouse is contain the human genes. If this virus, release to outside and just flying in the air and infected with human, how do you think? What's going on? Okay, so simply just I explained the data about this one, but how do you think? Why also isolated these virus from pangolin? Yes, just this virus stay in nature, so they stay in the pangolin. Never conducted with the human, but they try to transfer these virus in the laboratory and culture them, and they try to infect the mouse, human gene containing mouse, and carried out the pathogen challenge test, and all mouse died. So firstly, I said that when we carry out some experiment by using biochemistry or biology and new techniques, so at that time we have to think about the research ethics. Is it okay to carry out for human being? Yes. So that's why I selected this data. So you also, one of the researcher because you belong to the laboratory now. Okay, so after that, please check the guideline for mini report, and please supply your opinion from the viewpoint of the ethics or research philosophy. Okay, thank you.